Log on, tune in, find out. Another good idea from Cambridge.
Well, a warm welcome to this seventh lecture in the Darwin College lecture series on life. I assume you know you've come to life as a cook. Here was one I produced earlier. No, of course, actually the title is Artificial Life. My generation was brought up on Dan Dare, but subsequent generations were brought up on R2-D2 and Daleks and things like that. And these were largely concerned with mimicking humans. We're moving now into the far more scary world in many ways of synthetic biology, reprogramming living organisms. And the lecturer is Chris Bishop. He's a professor of computer science at Edinburgh. He leads the machine learning and perception group of the Microsoft lab here in Cambridge. And like four of his predecessors in this series, is a fellow of Darwin College. Chris was trained as a theoretical physicist at Oxford and did his doctorate at Edinburgh, and he worked on nuclear fusion for many years before moving on to work on neural networks and pattern recognition. To some of you, he'll be known for his textbooks on pattern recognition, but for many, many more of you, he'll be known for his wonderful Royal Institution Christmas lectures of, I think it was 3 years ago. He's Vice President of the Royal Institution, and he's had a number of awards for engaging the public in the excitement of science and engineering. It's a great pleasure to introduce Chris Bishop to talk about artificial life.
Thank you. Living organisms are extraordinary things. They have capabilities which far exceed any technology that we have today. And so it's natural that scientists and engineers will seek ways to emulate some of those capabilities in artificial systems, and that's really the subject of this lecture. And during the course of this lecture, we'll uncover some relationships, some very deep relationships, between two fields which at first seem to be poles apart: biology and computer science. Now, there are many aspects to artificial life, and in this lecture I've chosen to talk about three of them. So in a sense, this lecture is in three parts. And one of the things that's going to link these three parts together is this man, Alan Turing. Now, Turing was a Cambridge mathematician, and this year, 2012, is the centenary of his birth. So I thought it was quite appropriate that in this lecture we could celebrate some of the many achievements and many contributions that he's made to a whole range of different disciplines. Now, Turing is probably most famous, I think, amongst the general public for his work during World War II at Bletchley Park for deciphering cryptograms and, in particular, for breaking the Enigma cipher. And this is the Enigma machine that you see here. But Turing was also very interested in biology, and towards the end of his life, he started to explore questions around the mechanisms of pattern formation in nature. So here we see this beautiful cheetah. It has spots on its coat, and the fact that it has spots, of course, is determined genetically. But the individual spots are not programmed genetically. The genetics do not specify the size and shape and location of every spot. Instead, they seem to say that the coat should be spotty. It specifies spottiness, but not the details. The spots seem to arise in some sort of in spontaneous way that hasn't been well understood for a long time. And in 1952, just 2 years before his very untimely death, Turing published a paper called The Chemical Basis for Morphogenesis, in which he proposed a mechanism for the formation of patterns and structure in natural systems, such as the spots on this cheetah. So I thought we'd begin the lecture by looking at some examples of spontaneous pattern formation. This is also known as self-organization. So what I'm going to do is to show you some practical examples of self-organization. And some of these demonstrations are a little bit small, so we've got a camera, and hopefully we can bring this up on the screen, and you'll be able to see these in action. So the first example, and I'm putting on gloves because it's kind of messy stuff, is called a ferrofluid. So a ferrofluid is really a dark, oily liquid, and it contains in suspension billions of tiny magnets. So think of these as little bar magnets. And bar magnets, of course, left to their own devices, like to point in opposite directions, with the north pole of one near to the south pole of another, because that's the sort of minimum energy configuration. So inside this liquid, we have a sort of soup of these little bar magnets all trying to orient themselves in opposite directions to each other. And it just looks like a dark, oily liquid. But something interesting happens if I bring up a strong external magnetic field. So if I bring up a powerful magnet, you can see, hopefully, that something interesting starts to happen. What we see is the spontaneous formation of a pattern, a structure, a spatial structure. So we're seeing these spatial oscillations, these little spikes that are forming. What's happening here is those little magnets, when the strong external field appears, they want to orient along the direction of the field. But that means they're then parallel to each other. And they don't want to be parallel to each other. So they reach a compromise. They form these spikes in which the little magnets can align with the external field, and that the separation between the spikes helps the individual magnets to get apart from each other. So the liquid doesn't have any spikes, and the magnetic field just varies uniformly, but it's the interaction between them that causes that regular spatial oscillation to appear. Okay, so here's a second example. This is a very simple one. You can try this at home if you like. This beaker contains a mixture of ordinary white granulated sugar and a sand. And the sand is from one of these aquarium shops, so it's been stained black. So the granules of sugar, relatively speaking, are quite large and quite sort of angular, and the sand grains are quite small and quite rounded. Now, I've mixed these together pretty thoroughly. What I'm going to do is to pour them through this funnel into this apparatus, which just consists of 2 sheets of acrylic separated by about 5 millimetres or so. So let's see what happens if I just steadily pour this mixture into the funnel. So as you can see, it forms a heap, a little sort of pyramid, a sand pile. And the sand pile gets steeper and steeper until it reaches a critical angle. And then, as you can see, there's a little avalanche. And that little avalanche causes a separation between the grains. So we're getting these periodic avalanches. And I think you can see that this is leading to the formation of a stripy structure. So we're seeing this regular stripes, and each of those stripes, the black and the white grains have become separated. So again, this is an example where we have the spontaneous formation of a spatial structure. In this case, again, a spatial oscillation in the form of these stripes in the sand pile. Okay, so in the third example, we're going to do something a little bit different. It's a little bit of chemistry. What we're going to do here is to take a solution of some hydrogen peroxide, and this is just a magnetic stirrer. This just stirs the liquid. And we're going to add a solution containing potassium iodate and a little bit of sulfuric acid. And then I'm going to add a third solution. This has malonic acid and manganese sulfate. It also has a little bit of starch, which is just giving it that slightly murky look, and that will just enhance the coloration, as you'll see. And just watch what happens as I mix this third liquid in. So it starts off colourless. It's turned an amber colour. And we'll keep watching. It turns a very dark blue. Well, let's keep watching. Let's see what happens. And after a few moments, it goes very briefly to colourless and then back to amber again. And we'll keep watching. Any second, back to blue. So again, we have the spontaneous formation of a structure, but this time it's a temporal structure. We have oscillations through time. So we've seen oscillations through space, oscillations through time. And this particular reaction, or type of reaction, will play an important role later in the lecture. We'll come back to that in a moment. I'm just going to show you one more demonstration. I have to confess that this final demonstration doesn't really have much to do with the rest of the lecture, but it was too much fun for me not to show it to you. It has another benefit. It involves bringing some green slime to life, and I thought a lecture on artificial life, we have to have green slime coming to life. So the idea of this is quite simple. The beaker contains, again, this is an experiment you could do at home. The beaker just contains a mixture of ordinary cornstarch, corn flour, with water. So it looks like a slightly viscous liquid. It just runs around the beaker quite nicely. But if I try to move it very quickly, if I apply a high shear stress, if I hit it hard, it behaves much more like a solid. So when it moves slowly, it behaves like a liquid. When I hit it hard, try to move it, I can break it apart, it behaves like a solid. So I'm going to pour a little bit of this liquid onto this membrane. And this membrane is just sitting on top of an ordinary loudspeaker. And I've connected the loudspeaker up to a signal generator. So we'll switch this on. I'm driving this at about 30 hertz with a sort of a triangular waveform. And we'll see what happens as I turn up the amplitude of the oscillations. So we see some ripples forming. These are called Faraday waves. It gets really interesting if I crank up the amplitude a little bit more. We start to see some interesting patterns emerge. The projector has a hard job keeping up with this because it's happening quite quickly. I think you can see, certainly very apparent here, it almost looks like it's come to life. These sort of writhing snakes curling over each other and falling back again. So just a lovely example, I think, of spontaneous pattern formation. Right. Okay, so the key demo there is the oscillating chemical reaction there, the stirred reaction. That's an example of what we call a reaction diffusion system. And I want to show you, if we just go back to the laptop, show you another example. This is one I prepared earlier because this one is a relatively slow reaction. It takes 10 or 15 minutes, so it's too slow to show you in real time. So I've made a little video, and then we'll play this as a sort of time lapse. But it's a similar but slightly different chemistry to the one you saw in the stirred reaction there. What I've done is mix some chemicals together to make this red solution, and it's being poured into a Petri dish, and then splashed around to make it into a nice thin film. You can see it's changing from red to blue, back to blue to red again, so this is sort of already starting to oscillate. But this time, instead of stirring it continuously, we're going to make a thin film and then just leave it. And now the video is being sped up by a factor of 64 times real time, and we start to see some colour changes. But what's interesting is that not only is this oscillating through time, but it's also oscillating in space as well. So if you focus your attention on a particular spot within the solution, you'll see it oscillates red, blue, red, blue. So it's just oscillating like the previous reaction. But now it's organised itself into a pattern in space and in time. Okay, so that's an example of a reaction diffusion system. So what is a reaction diffusion system? Well, reaction, that's a chemical reaction, that's sort of molecules breaking apart and the atoms are rearranged to form new molecules, and we see the chemical reaction is expressed through a change of colour. So that's the reaction part of it. What about diffusion? What is diffusion? Well, imagine we looked down a microscope and we saw the molecules. What we'd see is that the molecules or atoms are in constant motion. This is thermal motion. So heat is just the continuous random motion of atoms and molecules. Now, each molecule is equally likely to move to the left or to the right. But supposing we have a concentration gradient, that is, we have a high density of molecules on the left and a low density on the right. Well, each molecule might move left or right with equal probability, but because there are more molecules on the left than on the right, then on average we have more molecules moving from left to right than move from right to left. It's just statistics. So that's diffusion. It's the sort of random motion of these molecules, on average, down the concentration gradient. Just switch that off. Okay, so that's diffusion, and Turing's mechanism is based on the interaction between reaction and diffusion, and his mathematics describe the formation of patterns in those types of systems. And this picture is taken from a recent review article by Kondo and Mura, and it shows Turing's main result, which is that there are 6 classes of solution, six phenomena that can be expressed through these reaction-diffusion systems. So here are the 6 possibilities. So the first possibility, and by the way, which of these you have depends upon the parameters, depends upon the rate of reaction and the rate of diffusion and so on. So the first possibility is that it's very boring. It sits there and does nothing. The second possibility is the whole system oscillates together. So that's rather like that stirred reaction we saw. It's oscillating, but it's all oscillating together. There's no spatial structure. There are a couple of sort of pathological solutions. We have very short wavelength effects. Those are not of interest to us. The fifth class is where we have oscillations with finite wavelength in both space and time. So we have these travelling waves. So the video I just showed you was an example of class 5. And the class that Turing was interested in, class 6, the so-called Turing patterns, are where we have a static spatial structure with a finite wavelength. So these might be the spots on the skin of a cheetah, for example. Now, what's been realized is that we don't necessarily have to have literally a reaction-diffusion system in order for Turing's mechanism to operate. We have to have a condition something like the following. So if we move now to a slightly more biological context, imagine that this is a cell. Well, suppose the cell could be one of 2 colours. And we'll suppose that we have several of these cells, and they can communicate with each other through chemical signaling. Now, if we have 2 mechanisms at play, so the first mechanism is a short-range positive feedback. So what that says is that if one cell is near to another cell, they should have the same colour. They tend to reinforce each other in having the same colour. But there's a second mechanism, which is a long-range negative feedback. That says if 2 cells are far apart, they signal to each other to have the opposite colour. Now imagine we have lots of cells. What's going to happen? Well, it's a bit like that ferrofluid. There's sort of one effect trying to make nearby cells in the same direction, rather like all the little magnets trying to line up the external field. And there's another effect, which is this longer-range effect, which wants cells to have opposite colours if they're far apart. And it ends up in a sort of compromise. And that compromise has these spatial structures, things like stripes and spots. So that's essentially how this short-range excitation, long-range inhibition mechanism can give rise to spatial structures, to Turing patterns. Now, here are some examples of computer simulations of the Turing mechanism. So in each case, on the left, we see the actual biological organism, and on the right, we see the corresponding computer simulation showing that the reaction-diffusion mechanism can indeed give rise to that particular structure. And we see a whole variety of structures have been simulated by the same mechanism. Now, that doesn't prove that this mechanism is responsible for those structures, but it does show that that simple mechanism is sufficiently flexible that it can recreate this tremendous diversity. And indeed, there is now quite compelling evidence that Turing's mechanism does indeed play a role, at least in the formation of some biological structures. And here's an example of some further verification of this hypothesis. This is, on the bottom we see a zebrafish. And at 13 days old, the stripes on the fish have been ablated using a laser. And then over the subsequent days, we see the stripes reforming. But they reform with this strange loop structure. And above is the computer simulation of the corresponding system, again showing that same loop structure. Now, we're going to return to Turing patterns a little bit later in the lecture, but what I want to do now is to move on to the second part of the lecture and to look at some other aspects of biological systems that we might wish to mimic in an artificial system. And I'm going to look at a topic which is very close to my own field of research. It's the problem of artificial intelligence. So there's no doubt that one of the most extraordinary capabilities of organisms, particularly higher organisms such as humans, is intelligence. And there's a tremendous interest in creating machines that exhibit intelligent behaviour. Now, Turing himself was very interested in this, and he grappled with the very slippery fish of intelligence, of trying to understand what intelligence is, by proposing something very practical. He proposed a sort of an operational test of whether a machine is intelligent. And so this has become known as the Turing test. And the idea is the following. We have some sort of a screen or a barrier, and on one side of the screen we have a person who's going to conduct the test. And on the other side we have the computer that's under test, and we have another person. And the person, the blue person on the lower side of the screen can ask questions of the computer and of the human on the other side. And generally this would be done through a simple text system. So there wouldn't be any voice synthesis or anything like that. It would be just simple text questions and text replies on a screen. And the goal is to see whether the blue person is able to tell at any time whether they're talking to the computer or to the human. If they can't tell the difference, then the computer is deemed to have passed the Turing test, and therefore, according to this specific definition, to be intelligent. I should emphasize, what the person here is not doing is checking whether the computer gets the right answer. So if this person, and they can ask any question they like, philosophy or talk about physics or anything they like, they could, for instance, give the personal computer on the other side a huge arithmetic cube root or something to do, and if the entity on the other side came back in a split second with the correct answer, they could be pretty sure that it's a computer, not a human, and the computer would have failed the Turing test. All right? So it's not about getting the right answer. Now, this is certainly very interesting, but there are some problems with this. So one of the problems is that this is quite a narrow definition of intelligence. So, for example, a 3-year-old child, certainly, and many animals, can recognize objects very quickly in the environment. Just looking around and saying this is a person, this is a table, this is a chair, and so on, is a very hard task, and is certainly an aspect of intelligence. We know that animals can recognize predators, recognize prey, and so on in a fraction of a second, and yet no animal could pass the Turing test because they can't mimic a human in this way. So, as one researcher said it, aeroplanes are measured by how well they fly, not by how accurately they imitate birds. So the real goal is not to pass the Turing test, it's to produce machines which have, one way or another, intelligent-like behaviour. But nevertheless, Turing's thoughts and work in this area stimulated tremendous interest, and it led to a field called artificial intelligence, which really flourished in the 1960s. And the main approach at that time was something called expert systems. And the idea was this. You would take a human expert, let's say you would take a doctor, a general practitioner, who would be good at diagnosing disease, and you would ask them how do they do it? How do they diagnose disease? And they would try to express their expertise as rules. So they might say, well, if the person comes in and they've got a high temperature and their blood pressure is normal, then maybe they've got a cold, and so on. So you would get all these rules out of a human, and you would put them into a computer, and you would run a special kind of engine so that when you gave the computer a set of symptoms, it would fire off all these rules and then come back and tell you what the disease was. So it was trying to mimic the human, very much in this sort of Turing style. And there were some early successes, and that led to tremendous optimism. The optimism in the field was quite remarkable. Marvin Minsky was really one of the fathers of this field, and he said, back in 1967, he said, within a generation, the problem of creating artificial intelligence will largely be solved. And the following year, Stanley Kubrick released a film based on a book by Arthur C. Clarke called 2001: A Space Odyssey, and it featured a computer in the year 2001, and this computer was intelligent by any manner of means that you could imagine. So that was the sense back in the 60s of where we would get to by the end of the millennium. Things didn't quite work out that way. I think it's fair to say, there are a few specialist situations in which this kind of approach does give you a good answer. If you want to glue two things together and you want to know what adhesive to use, well, there are too many materials for material A, too many possibilities for material B, and too many possible adhesives and temperatures and so on to list all the possibilities. But you can build an expert system that will solve that problem for you. But that's a very tightly defined specialist domain. In terms of producing machines that are intelligent in any general sense, this field was essentially a failure. And throughout much of the 1970s, 1980s, the whole field really went into decline, funding dried up, and so on. Now, the quest to build intelligence in machines really underwent a rebirth in the late 1980s, early 1990s. And this was because of a radically different approach. So in this approach, we don't program the computer with rules such that it mimics intelligence. Instead, we do something very different. We program the computer to learn from experience, and then we show it data. We give it experience in the form of data, and it learns from the data. So what I'm going to do is just show you a little toy demonstration, simple demonstration, which illustrates the idea of learning from data. So this is a little system. It's an example of what we call collaborative filtering, and it's going to learn about your preferences for videos or movies, films, and then it's going to make recommendations for other movies that you might wish to watch. So this system has been trained using data from tens of thousands of people with hundreds of thousands of ratings. So it's learned already from a large number of people the kinds of patterns of preferences that exist within the world of movies. What it doesn't know, it knows nothing about the individual movie. So each of these icons represents a particular movie, but it's not been told that this is a romantic comedy, this is an action thriller, and so on. It's just movie number 127. The only thing it knows is that people who like movie 127 also like movie 37 or whatever. So what I'm going to do now is tell it something about my preferences. So it knows nothing about me as an individual, but I'm just going to pick a film, pick a movie, and I'll drag it across into the right-hand column, and that says that I like that movie. And what's happened is all the other ones have rearranged themselves. And they're rearranged in a particular way. The vertical position on the page doesn't mean anything. They're spread out vertically just so you can see them. What's significant is the horizontal position. So the horizontal position is the probability which the computer has assigned to that movie that I will also like that movie. So in other words, if it's put a movie in the middle, that means it's sort of 50-50. It's unsure whether I will like it or I won't. The further it is to the right, the more sure it is that I will like that movie. The further to the left, the more confident it is that I won't like it. Okay? Now, to start with, they're all pretty much bunched near the middle because it knows very little about me except that I like Pretty Woman. That's all it knows. Right. So what we can do is give it a little bit more data. And as I give it more data, so here's a movie I'll say I don't like this one, you'll see they tend to sort of spread out sideways. So what's happening is the probabilities started out near a half, very, very unsure, and they're moving towards 0 and 1. They're moving towards certainty. The system is becoming less uncertain as it sees data, and that's the modern definition of machine learning, a reduction in uncertainty as the system sees more data. So we can give it another movie. Let's say I like this one. Let's say I don't like that one. And you can see I've only given it 4 data points, and yet already the movies are sort of crowding towards the outside, and the middle is now starting to look like a lot of white space. There are still a few that it's not quite sure about, but it's getting quite confident now. Let me show you one more thing. Let's suppose that I take a movie that's very close to the right-hand side. So this is one that it's very confident I'm going to like. And let me say, well, yes, I've watched that movie now, and actually I do like it. So watch what happens when I let go of the mouse. A tiny, tiny change. Because I've just told it that I do indeed like a movie that it's confident I will like. So I haven't really told it anything very surprising. I haven't really given it much information. If I take a movie on this side, so this is one that it's very confident I won't like. If I say, well, actually, I did like this movie. Now watch what happens when I let go of the mouse. It's a huge change, because there's much more surprise. In fact, mathematically, the formal mathematical definition of information is the degree of surprise. So in a mathematical sense, the first example on the right-hand side, I gave the system very little information by telling it something it already knew. In the second case, I gave a huge amount of information by telling it something that it found very surprising. So that's an example of a computer learning a skill, learning in this case to recommend movies to me based on data. It learned both from a big data set from tens of thousands of other people and from the few additional bits of data that I provided it with. So that was just a toy demonstration, but the software that's powering that is actually used in very large-scale practical applications used every day to make recommendations to tens of millions of people. So it's a very practical technology. What I want to do now, then, is to show you another example of this modern view of artificial intelligence, which involves learning from data. And it comes back to the problem I mentioned earlier, which is the problem of recognizing objects, this thing that the 3-year-old child can do with ease, just looking around the room and saying, that's a person, that's a table, that's a chair, and so on. It's a very hard problem, and by and large, we still haven't solved it. But we've solved some aspects of it. So about 8 years ago or so, in our research group here in the Microsoft Research Lab in Cambridge, we decided we wanted a new problem to work on, which would be very blue skies and extremely difficult, something that probably would be unsolved even in 30 years' time, but would really stretch our thinking in new directions. And we decided to focus on this problem of object recognition because we thought we had new techniques that could start to make a bit more headway with this. And I happened to be on holiday in Scotland at the time, and so we started to collect data, and the data we collected were pictures of cows and sheep on Islay, and that was the first data set that we looked at, and we collected other data sets around Cambridge, pictures of animals and bicycles and so on. And in this particular aspect of the work, what we're trying to do is not just recognize the object, but really classify every pixel in the image. So in this image, every little point in that image is classified as belonging to one of 3 classes. It either belongs to grass, cow, or water, and so on. And after several years of working on this problem, we got to the point where we could simultaneously address a few dozen classes and also do this processing really quite quickly. So the computer could, in a fraction of a second, take an image and label up maybe 20 or so different classes. So far, far short of what any 3-year-old toddler can do, but nevertheless a bit of an advance in terms of what computers could manage. Now, we did that because it was a challenging problem, and we never imagined at the time that it would have any sort of near-term practical application, and by a piece of good fortune, it turned out to be just the piece of research that we needed to solve a rather practical problem. So let me show you that. And it concerns a thing called Kinect. So the box here is a games console, and Kinect is the object that's sitting on it. And I'm sure most of you are avid computer gamers, but just in case not, I'm just going to show you a little video of Kinect and what it does. So this is somebody playing a game, and in this case sort of a whitewater rafting game. But what you see is that as she moves around, as she moves her body around, moves her arms and head and legs around, it's being tracked. That little avatar on the screen is mimicking her body motion. So Kinect is looking at her body motion and then tracking her skeleton, tracking the positions and angles of all the joints. So in this case, it's being used to control this particular game, but obviously it could be used in lots of different ways, as we'll see in a moment. And notice in particular that she's not holding anything in her hand. She doesn't have a games controller, and she doesn't have to wear any kind of fancy special suit with white blobs or anything like that, just walks in front of the sensor and then starts playing. I just want to tell you how this works. So the key to this is some technology that's a type of camera that sees the world not in color or in intensity, but in depth. So it's a depth camera, and it's based on 2 components, an infrared laser that throws out a pattern into the room, and then an infrared camera. And the way it works is actually very simple. Imagine this is our infrared laser, and it's throwing out some sort of structured pattern, structured light into the environment. And it's done in infrared because it means you don't have to have the lights on, and it doesn't interfere with... it just makes the whole problem a lot simpler to do everything in the infrared. And also, people can't see infrared, and so you can't see these patterns, so they're not distracting. So you throw out a random pattern, or a structured pattern, into the environment. If you were to observe that pattern from the same location as it falls on objects at different distances, what you would find from the geometry is it would always look the same. So if you project it onto a screen, as the screen moves further away, then seen from this position, just because of the geometry, it wouldn't change. But instead, if you view the pattern from some distance away, this is the baseline, and in Kinect, the baseline is maybe 10 centimeters. So it's a bit like the baseline between your 2 eyes. The reason you can see the world in 3 dimensions is because your eyes are separated from each other, and they get different views of the world. And in the same way here, if you view this from a short distance away, then if the structured light is projected onto a screen, if the screen is moved further from the laser, then the image moves to the right a little bit. So by looking at the position on the screen, you can measure the distance. let me just show you a little video just to make that clearer. So this is just a video of somebody holding up a piece of white paper in front of a Kinect sensor, and this is filmed using an infrared camera. And all they're doing is moving the paper towards the camera and away from the camera. And I think you can see that as it moves away from the camera, this pattern, this random pattern of dots moves to the left, and as the paper comes towards the camera, the pattern slides to the right. So if I just keep my laser pointer still, you see the pattern moves to the right as the card comes towards the camera. Now, this is what we call a pseudo-random pattern. It looks random, but we know the structure of that pattern, so we can recognize which little patch is which. And so by measuring its displacement, we get the distance. So what we've built then is a camera which can see depth. So it sees the world in 3 dimensions. So this is the video that camera would take. Each pixel represents not a colour or an intensity, but a distance from the camera. And we've coloured it so that the pixel is dark if the object is close to the camera, and it's light if the pixel is further away. So as this person moves their hands towards the camera, it becomes darker, and as they move away, it becomes lighter. Okay, so that's what the actual sensor measures. It's a video in which each pixel represents the distance from the camera. And because it's giving distance, we can project the same data from the top view or from the side view, and we're actually sort of seeing the world in 3D. And that's a big advantage. If you want to track somebody's skeleton in 3 dimensions, actually having a 3-dimensional camera makes the problem, I won't say easier, but a lot less difficult than using a regular camera. So that's why that's done. But the goal, of course, is not just to take this depth information. What we actually want is for the computer to recognize which is the right hand, which is the left knee, and so on, because it's actually got to track the whole skeleton in real time. And this problem was solved by appealing to that work on object recognition. So instead of treating this the way you might treat regular sort of tracking problems, instead this was solved in a completely different way by taking every pixel and recognizing which part of the body it belongs to. So we defined 31 different categories, each shown by different colours. So each category is a different part of the body. So instead of trying to tell the difference between a sheep, a cow, grass and sky, we try to tell the difference between the right hand, the left knee and so on. And we train the system using data, so it learns from data, as we've discussed, and this is the training data set that was used for this Kinect sensor. This is 1 million body positions captured in a motion capture suite, and we've sort of laid them out on the page according to size. So that's what the training data set looks like. That's 1 million images, 1 million different body positions that's used as the training data. And then this is the end result. So this is the video. This is a video as seen by the sensor. This is the raw depth information. This is what our recognition system is doing. So 30 times a second for every frame of that video, and for every pixel in that frame, it's saying, is that pixel a right hand, left knee, part of the head, or what? And it's actually assigning a probability, just like with the movie recommender, it's assigning a probability to each of those 31 classes. So what it's done here then is recognize the different parts of the body. And you can see that it's quite noisy. As she sort of moves a hand in front of a body or sort of bobs down or something, it all becomes quite noisy. So it's not getting every pixel right, but most of the time it's getting most of the pixels right. And that's enough to work out where the head is and where the hand is. And so that's what we have here. This is a front view showing the locations of these key body parts shown by these squares. And because this is a 3D camera, we have the information in 3D, we can view it from the side and we can view it from above. So that's an example of a modern, a recent application of machine learning to solving a problem which addresses a certain kind of intelligence. This is not intelligence in the sense of the Turing test, but it's building a machine that has the capability to make sense of the real world. And you saw somebody there using this to play a game, and there are lots of applications in gaming, but there are lots of other applications for the ability to track the human skeleton in real time using very cheap hardware. And one of the things that we've been exploring recently is an application in surgery. So when a surgeon scrubbed up, they're sterile, they mustn't touch anything. They can't touch a keyboard or a mouse to bring up the information on the computer, because that would destroy the sterility. They'd have to go out and scrub up again. And So generally what they do is they have somebody else drive the computer, and they just say, can you zoom in a bit and turn left and so on. That's obviously not very satisfactory. So we're using Connect to allow surgeons to bring up typically often three-dimensional medical scans, MRI scans and so on, take sections through them, zoom them around, rotate and so on. And that's all done with gestures. They just walk in front of the sensor and just make gestures. And that's undergoing clinical trials right now in UCL, and many other potential applications for that kind of system. So the key idea here then is not to program the computer with rules, but rather to have the computer learn from data, to learn from example. And this field is becoming enormously exciting. It's seen as sort of, possibly the next big thing in computing for a very simple reason, and that's what's sometimes called the data revolution. So there's a lot of data in the world, and there's more and more all the time. One study back in 2008 calculated there was 280 exabytes of data spinning on the world's disks. Now, an exabyte is 1 billion gigabytes. But what's really interesting is that it's been doubling every 18 months. And we say it's growing exponentially. So it's a very special kind of growth. Now, the digital revolution, which of course has transformed so many aspects of our lives, has fundamentally been driven by an engineering reality, which is that the number of transistors that you can pack onto a piece of silicon has been doubling every 18 months. It's been doing that for 40 years. That's called Moore's Law. And it's Moore's Law that's turned computing from, an aircraft hangar full of valves and costing a fortune to something that you keep in your pocket and more or less disposable. The same thing is happening with data. We're seeing an exponential growth in the amount of the world's data. It's doubling every 18 months, and it looks set to continue for many, many years. As more and more devices and sensors are built, and more of them are connected to the internet, we have the ability to collect ever-increasing quantities of data. And it's not just the amount of data, it's this connectivity as well. Much of this data now resides in large data centers. This is so-called cloud computing. Because devices are connected to the internet, we can draw the data together, and we can learn things by combining multiple data sets together, things which are not obvious in any single data set alone. And the potential applications of this are enormous. So I won't go into this any further now, but just sort of leave you with that thought that the data revolution is really driving artificial intelligence in a way that we hadn't really anticipated, certainly back in the 1950s. So that really brings me to the third and final part of my talk, and it's here that I really want to look at some of these very deep connections between these two fields which seem so very different, the field of biology and the field of computer science. And I'm going to begin with the thoughts of a biologist on this topic. And here he is, it's Paul Nurse. And about 18 months or so ago, he came and gave a talk at the Royal Institution called The Great Ideas of Biology. And he enunciated 5 great ideas of biology. He said the great ideas of biology are the cell, the gene, evolution by natural selection, life as chemistry, and the next great idea of biology, which is that biology is a system that manages information. Now, of course, a system which manages and processes information is within the domain of computer science. So I have to say it's very heartening when the Nobel Prize-winning president of the Royal Society tells you that the field of biology is really just a subset of computer science. I was quite pleased by that one. What about computer scientists? What do they have to say about this? Well, this brings us back to Turing again. Now, Turing, as already apparent, has made contributions to many different fields, but probably his most important contributions involved really laying the foundations of the field of computer science. And I'm going to tell you about one of his most important discoveries about computation. So he asked the following question. This is back in the late 1930s, before we had any real computers, but we can sort of cast this in the context of some computers. So here are some objects which are, in some sense, do computation. They process information. And he asked the following question. He said, is it possible to build a computer that's so powerful and so sophisticated that it can solve any computational problem? And he reached a rather surprising answer. He proved the answer is no. He proved that there exist computational problems which no computer can ever solve, in principle, ever. And let me give you an example. So an example he gave was called the halting problem. So it goes like this. I can very easily write a little computer program which, if I execute it, that is, if I run that computer program on a computer, it will run for a while and then it will stop. Then I just sort of print hello world and then stop. So that computer program halts. I can also very easily write a little computer program that will run forever. Line 1 might say print hello, line 2 says go to line 1. So it prints hello, prints hello, prints hello, goes on forever in an infinite loop. So that's a computer program that doesn't halt. So every computer program either halts or it doesn't. Now, if I've got a computer program that's a million lines long and is very complicated, it may not be obvious whether it halts or not. So I might try to get a computer to answer that question for me. In other words, I could try to write a computer program which does the following. It takes another computer program as input, and the output is to tell me whether that first program halts or not. So in the case of the print hello world stop, it would say yes, it halts. In the case of line 1, print hello, line 2, go to line 1, that runs forever, it would say no, this doesn't halt. He proved mathematically that you can never construct such a program. So if you said to me, you think you have constructed a piece of software which will tell whether any computer program halts or not, Turing can construct a particular program for which your software will either produce the wrong answer or it will itself never halt, it will run forever. Right? And therefore, if it runs forever, it hasn't solved the problem. So by feeding the software sort of back into itself in this rather ingenious sort of self-referential way, he gave a proof that there exist problems, there exist computational problems, which no computer can ever solve. So that was quite a shock. Okay, so there are problems that computers can't solve, but does that mean that some computers are better than others? Are some computers capable of solving a few problems, and better computers are capable of solving lots more problems, and really good computers can solve huge numbers of problems? Well, he also looked at this, and again he reached a rather surprising conclusion. He said that, obviously, we can build very, very simple computers. So a simple computational engine would be this 4-function calculator. It can add, subtract, multiply, and divide, and that's all it can do. So that's clearly a very limited computer. But if a computer has a little bit more complexity, it has enough complexity that I can actually program it, it turns out that what any computer can solve, any other computer can solve. So if we ignore some simple special cases, then all these other computers have the same computational capability. So the problems which any one can solve, all of them can solve, which any one cannot solve, none of the others can solve. And that's called universal computation. We sometimes say that a computer which is sufficiently complex to belong to this class is said to be Turing complete. Now, that doesn't mean that some computers are not better than others. You probably just went out last weekend and spent a lot of money upgrading your computer, and you're a bit cross now because the previous computer does the same thing. This doesn't say anything about how fast the computer is or how shiny and what lovely colours it is and so on. They can differ. Computers can differ in terms of what they can perform. They can differ in speed. And, of course, some computers have more memory than others. So if we ignore the speed, if we allow the computer to have as much memory as it wants, then all computers are equivalent. But, of course, some are faster than others. The computer on the right here is an interesting historical computer. It's called the Apollo Guidance Computer. It was built in the 1960s for the Moon program, and it was absolutely the state of the art. In the time it was built, most computers were occupied large buildings, and this was a very compact, very lightweight computer to go into spacecraft, and it was used to navigate the spacecraft to and from the Moon, and it played a very critical role in enabling the spacecraft to execute a soft landing on the Moon. So that's the Apollo Guidance Computer. The one next to it is the chip out of a credit card. You're probably all, you've got several of these in your pocket at the moment. And that, too, is a fully programmable Turing-complete digital computer. It's 30 times faster than the Apollo Guidance Computer. It has 100 times the memory, and sometime in the next 12 months, you're probably going to throw it in the bin and get another one. So computers have come a long way. So keep in mind that notion of universal computation. We'll come back to that in a moment. Let's have a look at the world of biology. And information in biology is understood through looking at DNA, and I'm sure you're familiar with DNA. DNA is this double helical molecule. It has two strands. The two strands sort of are complementary. They fit together. They sort of stick to each other, and they're wound up into this double helix. And this lovely video and some subsequent videos came from Drew Berry from the Walter and Eliza Hall Institute of Medical Research. And these are delightful videos. So let's have a look a little bit more at the structure of DNA. So we have these two strands, and each strand has this backbone shown by the grey blocks. These are sort of sugars and phosphates. And then attached to that backbone is a sequence drawn from an alphabet of 4 possibilities. So each of these so-called bases in this sequence is either cytosine, guanine, thymine, or adenine, C, G, A, or T. And so that's the sort of the alphabet of information storage in DNA. And DNA is a double helix, it's double-stranded, and the other strand is complementary. So any time that you see a G on one strand, it will always be matched with a C on the other, and if there's a T on one strand, it's matched up against an A. So the second strand is redundant. It carries equivalent information to the first strand. And just one more little technicality. Well, 2 more technicalities, really. First of all, these fit together. So the actual shape of these complementary bases actually fit together, and they form these Crick-Watson base pairs. And the backbones themselves have a directionality to them, and the direction for the 2 backbones is actually opposite. Their directionality is sort of mirror image of each other. And that's important in DNA replication. So this video, if we bring the lights down a little, this video shows, more or less in real time, an attempt to produce an accurate computer simulation of the process of DNA replication. So the DNA strand is being fed in here, and it's being split in 2, and this strand here is simply being, so here's the DNA coming in, it's split in 2, and this strand is being replicated. And look at this extraordinary machine, right? This is... Each of those little blobs is an atom. So this is biology, but it's chemistry, but really it's mechanical engineering. Look at that. Phenomenal. So this strand, the DNA is being split in 2. One strand is being replicated, but the other strand is back to front. So it has to be replicated back to front. So it's being built back to front, and when a section is completed, it's stitched on, breaks away, and then the next section is done. So this section is being replicated back to front. Just extraordinary. And there are lots of other things going on as well. So there are mechanisms in there for correcting errors and all sorts of things. That's just a glimpse at the extraordinary complexity of biology. So one thing that's very obvious, and I like to think that it must have been sort of very obvious to Crick and Watson when they first saw the structure of the DNA molecule, is that it is a digital information storage system. That's quite clear. So it consists of an arbitrarily long sequence of these bases drawn from these 4 letters, this alphabet of 4 possibilities. And I can easily just translate them into zeros and ones, if you like. So using 2 bits, 2 binary digits, I can represent the 4 bases using ones and zeros. And so if you give me a DNA sequence, I could translate it into ones and zeros, or give me some ones and zeros, some binary, I can translate it back into a DNA sequence. And the human genome has around 3,000 million of these base pairs. That's about 750 megabytes, which is the equivalent of one sort of Hollywood movie, roughly. Okay, so DNA has some very interesting properties, and one of the interesting things is that we can take a piece of DNA out of one organism and put it into a completely different organism, and somehow it can still work. And this is really the field of genetic engineering. And this goes back to really the early 1970s, and has proven to be really very useful and is very interesting. This is an example of some damage that's been done to the leaves of a peanut plant by the larvae of the cornstalk borer. And the plant on the right has been genetically modified. So what's happened is a piece of DNA, a gene, which causes from a bacterium, which causes that bacterium to produce an insecticide. That piece of DNA has been taken out of the bacterium and transplanted into the peanut plant. And now the peanut plant, as well as doing all the things that peanut plants normally do, also creates this insecticide. And so the larvae come along, they take a couple of bites, and they drop dead, and there's a sort of dead larvae there. So I've called this genetic engineering in inverted commas because it isn't really engineering as we've recognized as sort of a mature discipline. This is simply taking a gene from one organism, transplanting it into another, and noticing that it still works. So it's an extraordinary activity, but it falls far short of what we think of as engineering. If we're to do real engineering, we need things like standardized parts. Right? An engineer constructs something, they don't build everything out of, iron ore. They go to a catalogue and they look up standardized parts. Those parts have to have specifications. They need simulations with which to predict the behaviour of the completed system. If you're designing a new airliner for Airbus or Boeing, it's no good doing it by trial and error to figure out how big the wings should be. You have to design it on a computer. You have to be able to simulate it, so when you build the first actual aeroplane, it just has to fly first time. Too expensive to do anything else. And you need the ability to design desired behaviours. You have to be able to say, I want the system to behave in a particular way, and then design it so that it achieves those particular aims. So that's why I've called this genetic engineering in inverted commas, and I'll come back to that in a moment. This idea of gene transplantation can be taken to extremes. In a sense, this is an extreme example. Craig Venter is a notable figure in this field, and he was one of the people who was involved in the first attempts to sequence the entire human genome. And recently he did the following. He took something called M. mycoides, and it was a very simple organism with a relatively short genome. And he took out the DNA and he sequenced the DNA. So this is a fairly well-established technology. Today you can get the entire human genome sequence for not much more than a few $1,000s. So he took the DNA out of this organism, sequenced it, and so what he has now on his computer is the sequence of bases, the C, G, A, and T. He then made some modifications to the sequence. Now, in a sense, they're relatively simple modifications. What he did is effectively inserted watermarks. He inserted extra letters in blocks into the sequence in a way that wouldn't affect the biology. So he put in things like some quotes from some famous people, and he gave this organism its own email address, for instance. He then took the modified DNA and then used some other technology, which is DNA synthesis. So you can get a machine which is sort of like, it's a bit like an inkjet printer, but instead of printing cyan, yellow, magenta, and black, it prints C, A, G, and T one at a time. So it prints out one base at a time, and you can assemble a piece of DNA. That technology is much less advanced than gene sequencing. It can only print out relatively short sequences. So this was quite a sort of tour de force to reassemble the genome of this organism. It took 20 scientists 10 years, cost something like $40 million. And when he finished, what he then did was to take a somewhat similar, but a different organism, M. capricolum, and he ripped the DNA out of it. So it's like a kind of a chassis, an organism with its DNA removed. And he took the synthetic DNA and inserted that into the organism. And when it grew, it very quickly changed to look like the original mycoides. And he called this Cynthia. And in some sense, I think, tried to suggest that this is a synthetic organism. Now, the notion this is a synthetic organism is not entirely clear, because after all, the key thing about the DNA is the information which it contains, and that information has simply been copied from a natural living organism. And furthermore, the DNA doesn't work on its own. It has to operate in some environment, and that environment was itself a naturally occurring living organism. So this is still quite a long way short of being a truly synthetic organism built out of non-living components. What it does show, I think, is the advance in the techniques, the technology for manipulating DNA, for reading it, for synthesizing it, and so on. We want to do something, in a sense, that's much more ambitious even than that, and we want to gain deeper insights into the nature of DNA and its role in biology. And so what I'll do now is just show you a couple more of these lovely videos which show you how DNA actually gets used in living organisms. And Ron Lasky in last week's lecture touched upon this to some extent, and I want to just go into a little bit more detail. So the DNA stores this information, and it's used in a process called transcription, where a molecule called polymerase goes along the DNA thread and reads that information and then uses it to synthesize a related molecule called RNA. So when it gets the signal to go, here it goes, it shoots along the DNA strand, and what you see pouring out here is this molecule, which is very much like one strand of a DNA. It's got an alphabet of 4 bases and one slight difference, but it's very similar to DNA. So it's sort of copying the information that's in the DNA. And that's called messenger RNA. And you see again this extraordinary machine. That DNA is spinning at the same rotational speed as a turbine in a jet engine. And this is a real-time simulation, and the RNA is sort of spewing out. and then that RNA finds its way out of the nucleus, and then another molecule called a ribosome picks up the RNA and performs another process which is called translation, in which it uses the information, that sequence in the RNA, to string together building blocks called amino acids to make another long-chain molecule, which is a protein. And again, you can see it sort of chunting along. It's taking little triplets of these. Each little triplet, a set of 3 of these bases, is red and codes for one of these amino acids, which is then bolted onto this ever-growing chain of amino acids, which is this protein. Okay, so what we have then, DNA is this, let's look at one strand of the DNA. DNA is this sequence of these 4 bases, and chunks of this DNA, called genes, code for proteins. And I must apologize to the biologists for the extreme oversimplification of this discussion, but it makes the essential point. So a piece of this DNA will code for a particular protein. So the polymerase transcribes it into RNA, and the RNA is then translated into protein by the ribosome. But it's more complex than that, because the difference between a brain cell and a skin cell is in the proteins that are actually expressed. Not every protein that can be made is made, and whether a protein is made or not and the rate at which it's made can be controlled. And it can be controlled by another part of the DNA called the regulatory region that's associated with this coding region. And so we can have feedback loops between RNAs and proteins which influence the regulatory regions and influence the extent to which these proteins are expressed. Now, I've shown the feedback loop here as within one particular gene, but of course what we actually have is feedback loops across many genes and across many different proteins, and so we end up with sort of network diagrams like this, which are just phenomenally complex, so involving many, many thousands of proteins. Some of those proteins are there for structural reasons, they're the actual fabric of the cell. Some of them are there as information processing elements. They're there to signal the formation or the switching off of other proteins. So Michael Acomb, in the first lecture in this series, sort of drew an analogy between the biochemistry of the living cell and a microprocessor. We see this as actually a very deep analogy. So the DNA is more than just an information store. It's part of a computational engine. And it's been proven mathematically that this is Turing complete. It's a universal computer. So every cell of your body is capable of the same computation, and it has the same computational limits as your cell phone or your credit card or a supercomputer. Of course, biological computers, like other computers, can differ in other respects, not in terms of what they can or can't compute, but in terms of their size and their speed and so on. And biological computers are really quite remarkable. So they can store a gigabyte of information in a millionth of a cubic millimeter. They're massively parallel, which means they're performing lots of operations at the same time, and that's just in one cell. Of course, there are huge numbers of cells in your body, as you heard last week. They're robust to failure. They're self-repairing. They have an incredibly efficient power supply. They do a lot more computation per unit energy than our best silicon computers do today. And of course, the most amazing thing is we don't have to make them in a factory. They make themselves. Okay. So our silicon computers have got a long way to go before we catch up with biology. So we have these complicated networks, and these networks form a Turing-complete digital computer. And so if you're a computer scientist, your natural inclination is to want to program or to reprogram these biological computers. Now, if we're going to reprogram the biological computers, because they're so complex, we're going to need tools. Just in the same way that when somebody writes software for a digital computer, they don't write machine code by hand, they're low-level instructions. They use tools to help them write the software. We're going to need tools. We're going to need tools that will take a given network system and simulate its behavior, as we talked about with the aircraft, for example. But also we need to design the system. We need to be able to specify a desired behavior in a living organism and then work out what DNA sequence we need, what software do we need to run in order to achieve that desired behavior. So let me show you a simple example of some work that's going on in this area. And this is work that's being done by Andrew Phillips and others who are colleagues of mine in the Microsoft lab here in Cambridge. So this is a very simple example, first of all. This is a detector. So the idea is that this is the specification for what we want the cell to do. So we want it to detect a signal, and it should produce some sort of protein that's a receiver that will detect this signal, this other molecule, and when it binds to the signal, it will trigger the creation of some protein. And the protein is called green fluorescent protein. It's very popular in this field because it's a protein which, when you expose it to ultraviolet light, glows green, and so you can very easily detect whether it's being produced or not. And therefore, it's a very simple marker that you can use as sort of an output device for your computer. So that's the desired behavior, and we can express that as a little piece of computer code. So here's the computer code that expresses this desired behavior. That can then be fed into this piece of software called GEC. And what this software does, it has access to a sort of database of biological parts. And it knows the behavior of each of those parts. And it can explore lots of combinations until it finds a solution to this problem, a system which exhibits the desired behavior. And it can then it can simulate that solution. So this is a signal is received, and this curve shows the production of the green fluorescent protein in this simulation. So having simulated the system and discovered the required software, we can then go ahead and build the system, and this has been done by Jim Hasselhoff and colleagues in collaboration with Andrew Phillips. Jim is in the Department of Plant Sciences here in Cambridge. And so on the left, what you see is a computer simulation of a colony of cells that have been programmed in this way. So imagine these are in a Petri dish, and we put the signal molecule down in the middle, and what we see is this wave of expression of green fluorescent protein spreading out. So that's the computer simulation. And the video on the right is a video of an actual colony of E. coli that have been modified using this desired program and exhibiting very similar behavior. And so we're actually testing the computer program in a living system. And that of course, can be used to verify the correctness of the software and to calibrate it and so on. So that's a very simple example. Let's look at a slightly harder problem, and I'm going to return back to those Turing patterns again. So let's see if we can program a cell to form Turing patterns. So this is the specification. So again, the cell is going to produce green fluorescent protein, but it's also going to send out two signals to other cells. One of them is a short-range activator, and when the cell receives this activator, that will stimulate the production of the protein. And then there's a longer-range inhibitor, and that has the converse effect. That shuts down the production of the green fluorescent protein. And if we simulate the behavior of a colony of these cells, then this is what we see. We see the emergence of Turing patterns. And this circuitry has been implemented, again, by Jim Hasselhoff in organisms, and this is in the process of being tested as we speak. Now, this really is a field that's rather different, I think, in spirit from traditional genetic engineering. So genetic engineering, you take genes from one organism, transplant them into another, and the new organism exhibits some of the characteristics of the old organism. This is really trying to do true engineering of biological systems. And it's remarkably accessible, not only to professional scientists. So if you go to a web browser and you search for DNA synthesis, and you click on a link, you can go to a site where you can simply type in a sequence of bases, C, G, A, and T. And, of course, you also have to type in your credit card. It's about 30p per base, I believe, for the price is falling all the time, of course. And you click the order button, and a couple of days later, a FedEx package arrives, and you open it up, and inside of these little bottles containing those DNA sequences. And in case you're wondering, I should say that they don't just ship to private individuals, only to bona fide users. And they do search, if you type in a sequence, there are searches done to make sure they don't contain pathogens or toxins. But nevertheless, this is remarkably accessible. And in case you don't know what sequences to type in, well, there's another project which is called BioBrick, started by Tom Knight from MIT. And this is sort of an open source project to gather standardized components for doing synthetic biology. And there are about 5,000 standardized components available on this database. And again, you can try it. Just go to that web address, look up these components, find one that does something interesting, click down a couple of layers, and you'll actually find the DNA sequence corresponding to that component. And so it's not just professional research scientists who are experimenting in this field. There's an annual competition called IGEM, the International Genetically Engineered Machine Competition, which is an undergraduate competition. These sort of cross-disciplinary teams of biologists and engineers and computer scientists and social scientists get together and enter the competition. They design some organism that does something interesting, and there's a competition. This is the 2010 photograph showing all of the participants. This competition has its own sort of Moore's law. It's kind of doubling in size every year. And in 2011, they actually had to run multiple competitions around the world and then bring the finalists together for a final sort of World Cup final. So lots of different projects. An example of a project from Cambridge is E. chromi, which is a modification of E. coli, which changes one of 5 different colours when it detects the concentration of an environmental toxin. And in the US, there's even a movement amongst high school students. There are sort of after-school clubs where the high school students get together with their teachers and they synthesize new organisms. But of course, it's not just student projects. There are also very serious, very large-scale projects. Jay Kiesling from Berkeley, for example, has created a new form of yeast using 10 different genes from 3 different organisms. And this yeast synthesizes an important anti-malarial drug called artemisinin. And it's an effective treatment for malaria, but it's very expensive because it's extracted from natural plants. And producing this from yeast could reduce the cost by a factor of 10. So that's a very exciting project. And there are many, many others. So this is, in a sense, a new field, synthetic biology. It aims to go well beyond the aims of traditional genetic engineering. And the question is, what will the practical implications of this be? Well, there was recently a report produced by the Royal Academy of Engineering, a relatively sober, learned society, not prone to exaggeration, which was really very bullish about the potential impacts of this field. It likened it to the transformation in the field of chemistry in the 19th century, when chemistry moved from an observational science to the field of synthetic chemistry. And that had enormous impact on fertilizers and food production and petrochemicals, the creation of plastics, and of course the enormous impact on the pharmaceutical industry. So today we live in an unsustainable world. It's based on technologies which involve the one-way consumption of finite resources. We urgently need to move to a sustainable world based on renewable resources. And for the most part, renewable resources will mean biologically based resources. So the ability to reprogram biology with the help of tools and insights from computer scientists will, I think, be hugely valuable in that endeavour. Thank you.
Well, we really have learnt how the leopard got its spots, or rather how the green slime can be taught to dance. Extraordinary explanatory power of treating information in terms of probability. I think I come out of that thinking that it's not a question of how computers mimic us, but how we're all mimicking computers, and the rather disconcerting implication that we're all inherently reprogrammable. Chris, that was a wonderful insight into where computer science is taking us with all its ethical, political and social implications. Thank you very much indeed.